kind of diagrams right here at the beginning of this box. So that the box represents a place where there is DNA sequence coding for amino acids not interrupted by stop codons until we get to here. Then we get a stop codon and that ends the open reading frame. And that would signal the place where the conclusion of the protein sequence would be. Then we have, what else do we have here? Promoter. Where's that going to be? Before the ATG naturally. Enhancers, where would they be?
Anywhere.
Pretty much anywhere, yeah. It could be, usually we think of them as being more 5' or more upstream here, but in fact they could be downstream, they could be in introns. Maybe not how it's shown you in introns in this particular case. Okay. So that's a little bit on gene structure. Okay, then we get to an actual example here where we have two HOX genes, and their names are HOX B5 and HOX A5. And notice that they start with methionine. That's your first amino acid in the open reading frame. And then we're looking at an alignment here with the homeodomain shown in gray. So the first question was, what does it mean to say that a protein is conserved? Who would like to define that?
Well, it's evolutionarily conserved, which means it probably has very little variation at all, if any at all, within the species.
Okay.
Also very low variation in other families.
Okay. So looking at species, different species in evolution or in families, we see that a gene is conserved, there's variation. Okay, great definition. How many amino acids in this homeodomain? Just count them up, how many did you get?
I just rolled a 60 on your count.
You knew already, without even counting. 60. That's because I told you.
Yep.
Right, so if you want to see, if you want to actually count there are 60 of them, and that portion of the protein is the homeodomain. You know, there are other portions of the protein that are not part of the homeodomain. I just point that out because that was confusing in the past. So then if we look at the sequences in general compared to the homeodomain per se, and we think about conservation, how similar it is, what is the difference here?
They're not very similar.
Where are they not very similar?
Everywhere outside of the homeobox, the homeodomain.
Okay, so not so much similar outside of the domain, but within the homeodomain, very highly similar. And you know, these two genes are not entirely unrelated. So you can find some places like they both start out MSS, Y, F, B, N, S, etc., etc. So there are other little places within the gene that are sort of similar. And you know, this is in general true throughout the gene. And if you did a comparison and you put a number to it, it might be something like, I don't know, 45% identical. That's how many matches you would find from one amino acid to the next. Then if you looked at that and asked how many matches, exact same identical matches within the homeodomain, you find over 90% are exactly the same. Okay. This happens to be two different homeobox genes within one species, but you could also look between species and you would find still that in particular this homeodomain region is going to be the most highly conserved part of the gene between species or among species. Yeah?
And it has to be, you said like, does it have to be 90% conserved to be considered then a homeobox gene? To have that sequence, have to have 90% of that sequence
You have to have
in a gene?
You have to have this sequence here, the 60 amino acids, in some kind of recognizable form. It may not be exactly 90%, and depending on what your comparison is, say if you're looking at a fruit fly versus a mammal, maybe there's slightly more variation compared to a mouse and a human, but it's going to be recognizable as a homeodomain. Yeah.
So it's that same sequence then that's across all different kinds of taxa. So there isn't like multiple different homeodomains. There's just the one that we see in all these homeobox genes.
There's pretty much just the one,
Okay.
which, you know, I'm not saying it's 100%, but it's
Right.
you know, recognizable. So within mammals, there's going to be something like 42 42 different homeobox genes. So 42 totally different genes, they all have that part.
Okay.
A fruit fly has them, a sponge has them, a lobster has them, and they all have, you know, you could recognize that part of the gene. Okay, so this is, we call this the homeobox if we're referring either to DNA sequence or homeodomain if we're talking about protein. And you'll see sometimes other types of boxes, like the T box or a paired box or these different terms that end in the word box. Those also refer to conserved domains. And it's the same sort of idea as there's a little portion of that protein that you can find as a recognizable tag in all the family members for that particular family. But the homeobox is sort of special because it's one of the most highly conserved of these little sequences among any gene family. Okay, so here's an example of a MADS box. So that's another conserved region. There are several different genes and a variety of species that have these. Actually, it's mainly plants that have MADS box genes. And the question was, how long is the MADS box? And I actually don't think the answer up there. So just by chance, it turned out also to be 60 amino acids. Not every case is this box region exactly 60 amino acids. Some are longer, some are shorter. And then, is the MADS box the same amino acid sequence as the homeobox? No, right? So this is where I'm saying that you just need to see this for yourself, and see actually the amino acids written out. You can tell that they're totally different and unrelated to the homeobox. But among the MADS box family, they all have this recognizable tag. And then elsewhere in the protein, they're not so similar, but within that MADS box region, they are. Yeah.
So in the homeobox genes, the thing that distinguishes them is that they have the homeobox in them. And it's not that they share some kind of general function in the different animals. So I guess what I'm trying to say is, are there other similarities between homeobox genes other than the homeobox itself?
That's a great question. Are there other similarities among the homeobox genes beyond the homeobox itself? And yet, you could make an argument that they have a similar function. So they have a similar biochemical function. They're all transcription factors.
Okay.
They have a similar developmental function in that they're expressed in these discrete places, and they help specify body identity.
Okay.
So, one might specify the head, a different one the tail, so their individual identities are not the same, but they have that common theme of this is what I do in life as a gene, and this is how I do it as a transcription factor. But the criteria, the fundamental criteria for getting entrance into the homeobox gene family is that you have to have a homeobox. You have to have that particular sequence. Other thoughts or nuances here? Okay, so now then we flip over and start looking at some phenotypes. And this gets back to the idea of homeotic transformation. And I give you a plant example, and it happens to be a MADS box gene that's mutated here. And so, what is the difference between this wild type plant and the mutant plant in cell 30? What got changed?
Stamen got turned into a purple, and petal got turned into a sequel.
Right. So a stamen, which should have been layer 2, got turned into a carpel. And a petal, which should have been the 3rd layer, got turned into another sepal. So actually 2 different body parts got converted to something now different within the body. And this is a different example than the fruit fly with the wings. But instead of just a crumpled up petal or a non-functional petal, we now got instead an entirely different structure, a sepal, that is intact and recognizable. So now we have here 2 examples where we have a plant example, one part changed to another, a fruit fly example, one part changed to another. But then we look at this 3rd example, which is a T-box gene. So there's a recognizable conserved sequence known as the T-box in zebrafish. And when you look at the phenotype of that, they're missing the pectoral fins. So here are the pectoral fins in the wild type, and in this mutant, they are just gone. So, the question is, the first two are called homeotic transformations. This T-box gene is not a homeotic transformation. Why is it not a homeotic transformation?
Because it's just a deletion of part.
Okay, it's just a deletion. It didn't change the identity, it just made it defective, or in this case, made it missing. Which, you know, in the interest of full disclosure, there's not that many really clean phenotypes that would just delete a body part. Usually it's still there, like a stumpy, malformed fin, but this was a kind of a cool example because it was just okay. So is this clear what's a homeotic transformation versus just a regular old gene phenotype? Okay. All right. Good. So hopefully that will help. If you want to pass these over this way, select them.
So with that, the homeobox gene, so if they go in there and gene target and make mutations in the homeo domain part, is that going to just knock out the function? Because it seems like you could argue that the homeo domain that gives those genes its function.
Okay.
I'm sure we'll run experiments.
I like that question. So the question is, if we did a mutation, if we had a mutation, if we engineered a mutation in the homeobox region, and we interfered with the homeo domain, would that be enough to block the function of this gene? How many think yes? How many think no? How many not sure? Okay. I think the answer would be yes. Because if you don't have the homeo domain protein, if you don't make that portion of the protein, you're not going to be able to do your job as a transcription factor, which is to recognize your target, because the homeo domain touches the target portion of the DNA. If you can't do that, you won't be able to turn on your downstream targets, and therefore you're never going to be able to accomplish the normal function of this wild-type protein. Does that make sense? So you could maybe mutate down in the, read prime end and just change some other domain and still retain a lot of functionality, but maybe not quite perfect for that protein. But if you interfere with that critical homeo domain, which after all is the most highly conserved because you can't tweak it that much and still get function in evolution, then you lose the function. Another question?
No, I'm just kidding.
Okay. Just kidding.
That was just so you could argue things that are, the more highly conserved something is, the more important it is.
Yeah, the more highly
That's the whole-
conserved is more important, kind of by definition,
Yeah.
because evolution is experimented. It's tried to tweak this, and it can't get away with it. So it's been forced to maintain this particular combination of base pairs and amino acids in order for the organism to survive and reproduce and get that gene to move forward in evolution. Okay. All right, so let's go then back to this slide here. And this is to talk about another principle that's associated with these homeobox genes in the fruit fly. And it's kind of odd, and I mentioned this briefly before, but all of these 8 genes arranged here on the chromosome have a certain order in the genome from 5' to 3'. And 5' to 3', that orientation is just based on the DNA structure. You know, which side had the OH hanging off the end, and which side, you know, how the sugar phosphate backbone is oriented. And so these genes are on here in a particular orientation, and if you look at where they're expressed in the embryo, the ones that are the most 3' on the chromosome are expressed the more anteriorly in the larvae. And then each of these different regions in the larvae corresponds to an adult region as well. So the red region here is going to correspond to the head, and then down here blue and dark green corresponding to more of the thorax and abdomen. And so this principle is called collinearity because there's this correlation between the order in the genome, the region that it's expressed, and then each of these regions develops to unique portion of body identity. So I'll just point out also that some of these genes are expressed in big swaths. So this purple gene goes all the way from thorax clear down to the abdomen. So they overlap a little bit. It's not like there's one gene only in that area, but you get a unique combination of these homeobox genes being expressed in each region on the anterior posterior axes. Okay, so that we covered. So then, what about in mammals? And it turns out that mammals do have a version of these 8 genes. As a matter of fact, they have 4 sets of them. So from this, we can infer, we hypothesize that that original region of the chromosome there might have undergone 2 rounds of duplication. The first round made 2 copies, and then somewhere along evolutionary time, that duplicated again. So now we have 4 copies. So we get 4 different chromosomes that have Hox genes on them. In addition to that, some of the individual Hox genes duplicated on a chromosome. And so this gave rise to now 13, up to 13 possible Hox genes located together contiguously on one of the chromosomes. And over time, some of these genes went defunct. So you'll see some are missing. So we have here an A2 gene, a B2 gene, but there's no C2 gene. It just became a pseudogene or got lost in evolutionary time, and same here. So this is sort of the snapshot in time that we're at with mammals right now, where we have these 4 different clusters known as Hox A, Hox B, Hox C, and Hox D, and they have up to 13 members. And all the genes that are represented here still have an intact homeobox that we could recognize. They're all functional genes that make protein products, and those products are all transcription factors. Moreover, the mammalian genes also show this sort of unique and special situation of collinearity. So their expression patterns in the embryo are within certain anterior posterior domains that kind of match up to their position in the chromosome. Now, Drosophila, when we looked at those embryos, we could really recognize these nice segments that were very chunky, and we could track out what they became. In mammalian development, you don't get, it's not insects, so you don't get the same sort of segments and parasegments and the division of the axes on that same methodology. But nevertheless, you still get these domains of Hox gene expression, and they still seem to correspond with this collinearity. So it's kind of neat that there's some features that are the same, but others that are a little bit different too. Then one other thing to point out here. Since we now have up to 4 copies of each of the Hox genes, you can begin to think about, well, how similar are these 4 copies in the 4 different chromosomes? And these can be referred to as paralogs, or paralogous groups, subgroups. So for example, Hox A1, Hox B1, and Hox D1 are all paralogs, and these would originally been all derived from the same ancestral gene. So as time went by and these whole clusters duplicated, these three have a gene, an ancestor gene in common. So you might expect that they're going to be more closely related to each other than they are to other Hox gene members on the chromosome, and that's actually true. So within these paralogous groups, you would see more similarity, not only in the homeodomain, but basically throughout the whole protein, because they're descended from the same gene. More similarity to each other than to other Hox genes in general. So paralogs to describe these closest gene within a species, and then homolog, it's more of a general term to just say that they're kind of related in sequence. Okay. So another thought to think about here is if these genes are very similar in sequence, maybe they're similar in function. Maybe they're similar in timing of expression and space of expression. And so if they're similar in function, they might be both actually doing at least partly the same exact thing in the embryo. So remember that idea, because it'll come into play. Okay, so here we are, we have some mammalian embryos and Hox genes are expressed. And here are some of the things that the Hox genes do in vertebrates. In flies, we said they're busy making thoracic segments and wings and so forth. In vertebrates, the Hox genes are really important for the shape and identity of the individual vertebrae. And this sort of makes sense because we're talking about the anterior-posterior axis here. And if you think of the structures that change and move, you know, our vertebrae are definitely an anterior-posterior sort of a thing. And each one of the vertebrae is a little bit unique and different. And I think, you know, doctors and vet students have to learn more about the morphology and the different shapes of each of the vertebrae and how that plays out down the skeleton. Central nervous system. So a lot of the different nerves, they have a very unique location along anterior-posterior axes where they need to originate and then branch out and go to different parts of the body. So you can't just take this one nerve and see it sort of slightly, you know, anywhere within a range. No, it's going to be on exactly the same spot from one individual to the next within a species. So that's something that has to be patterned on this axis. It has to be set up where this nerve is going to come and where it's going to go. And then also, what about limbs? Ribs, limbs, pelvis. These are always attached at the same spot, anterior and posteriorly as well, from one individual to the next. And you know, that doesn't just happen, it has to be told to the embryo, or the embryo has to somehow figure out, where should I grow the arm, where should I grow the leg, how am I going to evaluate where that should happen on this axis. And so the Hox genes, when we say they're involved in body identity, this is the kind of thing that we're talking about, is where are these structures going to develop on this particular body axis. Okay. So, and then that also makes these pretty interesting genes for developmental biologists, because this is the question we're trying to ask, is how do we get this final morphology, this shape, this number of limbs, and the Hox genes are part of the answer to that question in a really fundamental way. Okay, that brings us to the Hox code hypothesis, which just happens to be surrounded by 4 stars in a yellow box. So that might be a good test question. But the concept is pretty simple here, and it's basically saying that the Hox genes, not as individuals, but as different combinations, are going to collectively give each little region of the anterior-posterior axis a different message, so that it can be just like instructions for that particular region of the body to know what its identity is and to develop the appropriate patterning based on these genes. So we saw how many Hox genes there are. I should count them up and find out, but I think there's roughly about 40 or 50. And you know, these are not the only genes that are busy patterning on the AP axis, but they are major players. And you know, we already have this little tag to identify them by, so it's a nice starting point. Here's some of the evidence. So one of the things I like to try to do in the class here, since you're mainly seniors or getting close to being seniors, is to start thinking a little bit about how do we know the things that I'm telling you here, how do we know there's actually true? What kinds of scientific evidence would tell us this or support this? And one biggie is mutants. So if we can find instances where these genes are now not in their wild type form, what happens? And these mutant phenotypes can teach us a lot about the function of the genes and also about the mechanisms that the organism has for trying to accomplish these goals. So this is mouse knockout. It was one of our reverse genetic strategies where we could take a gene of interest. So we pick one of the Hox genes in particular, you go in and you make a mutation in that gene exactly, and then you see what the consequence is. And so by that kind of approach, you can learn a lot about what is the individual function for this Hox gene, or maybe mutate two at a time and see what happens then. And that provides a lot of support for the idea that these are patterning within their early embryo. Retinoic acid. So retinoic acid is a morphogen for mammals, and it's arranged in a gradient. It's a paracrine factor, so you'll find one portion of the embryo where there's really high levels of retinoic acid and then it tapers off. And the reason I mention it here is that one of the things the retinoic acid does is turn on or off target genes that are Hox genes. So one question is, well, the Hox genes do all this regulation. What regulates them? And part of the answer is morphogens within the embryo. And also, these are vertebrates we're talking about here, not fruit flies. And lo and behold, they're also applying the same sort of paradigm of a morphogen, concentration, and cell fate. And they're tying it into these key transcription factors, and that's how they set up their body plan, which is kind of a neat parallel between something with a totally different way of doing things, Drosophila versus humans, and yet you use some of the same tools. This is a fun one too, comparative anatomy. So if you look at the fish pectoral fin and the bat wing and the human arm, we'll get to this later in the course. So the fish pectoral fins are the set of fins that was mutant in the TBX5 mutants. They come off at about shoulder level. Structurally, the bones or structures within there are actually quite similar to the bat wing and to the human arm. So there's certain fundamental structures that you'll find in common among all these three things, even though one is a wing, one's a fin, and one's an arm. And they're all using some of the same Hox genes to set up and pattern these little structures of the cunning limbs. Also, you can think about things like snakes. So snakes have this huge, long anterior posterior axes with tons of vertebrae in there. And think about what the Hox gene expression might look like in a snake embryo. And maybe that Hox gene levels have been manipulated or changed in evolution, and now some of the Hox genes are expressed in really broad domains, and then they end up getting many vertebrae in that section of the embryo. So I think you might be able to envision how you could tweak and play with these Hox genes and where they're expressed, and then the outcome for body plan and morphology would change accordingly based on this information. All right, so in fruit flies, we said, if we mutate the homeobox genes, we can get a homeotic transformation. Could this actually happen in a mammal also? And mammals, for one, we don't have segments like fruit flies. For another, we got 4 sets of these guys, not just one. And another, we're mammals, and so is it even possible to get a homeotic transformation? And so to answer this question, researchers started going through and doing targeted mutagenesis, making mutations in individual Hox genes and analyzing the embryos phenotypically and seeing, well, okay, what happens here? Is it basically break it or convert it into something different? And one of the first genes that was mutated is this Hox A3 gene. So one of the things that happen if you mutate both copies in the diploid organism, the mice died soon after birth. So that at least tells you this gene is important for embryogenesis. If we don't have it, the thing is going to die. So it is probably doing something that's critical for making the body plan. Where is it expressed? Well, it's fairly close to the start of that Hox cluster, and in the embryo it's expressed pretty close to the anterior portion. So here's an embryo, and this is the head, and this circle here is the eye. This is the neck region where the pharynx is, and then you get down to the upper trunk and shoulders. And here in the pharynx or throat-type region in embryos, you can find these series of about 4 to 6 bar-like structures. So here's a cross-section through an embryo, and you can see here's one chunk of tissue. It's a bar, another bar, another bar, and another bar. So just the whole tissue of the embryo kind of gets into these mini segmented region right here, the throat area. And then in between each bar of tissue or each pharyngeal arch is a little region that is a pouch, or an area where there are very few cells developing. And these arches and pouches eventually down the line form very specific things for the adult organism. For example, pouch 3 here, that's going to become the thyroid gland, and pouch 4 becomes the parathyroid gland. And these are just 2 structures in the endocrine system that you typically find in this particular area of the individual. So, HOX3 happens to be expressed in about this region of the embryo where these pharyngeal arches and pouches are developing. And if you go in and look at the consequences of the homozygous mutation in HOX3, the structures in this area are the ones that are affected. And that basically makes total sense, because you would expect the region where it's expressed would be the region that might be altered or damaged if the gene is missing. But, so pouch 3